back. I always get frightened after the first exam that people are like going to result and not show up. So, no, they're not graded. I'm through a page so far. I'll have them done by the end of the weekend though. So you'll see those on Monday, and I'll probably just put together a couple of notes for Monday too to talk about them. Couple of things to announce real quick that are coming up. So one, you and your partner have to put together a table of the metabolic test that you did and what the results are for your unknown and the interpretation of that. That is for me to kind of do a quick glance and determine, are you on the right track or are there some bigger issues that need to be addressed? I don't think there are going to be any, in all honesty, but this is a kind of a quick assignment for me to make sure that when you go ahead and put together your video, there aren't any questions about what the results are. Okay? Erica, it is due Tuesday. And I think technically it's like due at the end of lab on Tuesday, so that folks who still have tests that they're finishing up can come in Tuesday and finish analyzing.
So is that, like, are we doing a lab on Tuesday?
Oh yes, we're doing a lab on Tuesday. On Tuesday we're going to start measuring growth of our unknowns. Yay! So basically everything we're talking about today, we're going to do an experiment with it on Tuesday. And that'll be the next comment is that'll be the last part of what you need for the video. So by Thursday next week, you'll have everything you need to put together your video with your partner on your unknown. Let's do 2 weeks from yesterday, just to give you a heads up. Okay. So next week we've got setup of growth. We'll analyze the growth on Thursday. The following Tuesday is a day for you to be working in the lab on your on your videos, peer reviewing with another group, and then hand them in on that Thursday. And then we're about ready to do a switcheroo and create lab groups of 3 and 4. We'll talk about that later. All right, Devin.
Do we not have lab? Not next Tuesday, but the following Tuesday.
So we'll talk about it. I have to double check the schedule, but my plan was to have it and have you guys come in and peer review with another group on your poster presentations. But I may change that plan. I haven't decided yet.
Is fall break though?
Oh wait, I'm sorry, not Tuesday. You're absolutely right. You're absolutely right. I apologize. Everybody's like, what the hell? Why is he forcing us to do this? I have to look at the schedule then, because I don't remember. I'll double check. Actually, let's just do that right now, because now I'm like anxiously fretting what the hell's going on. So
is it in
I don't know. Let me check. Oh gosh, I've been up since 4:10. Okay, I got here at 6 to start an experiment, because I love research too. No, not gross. Awesome. Grossly awesome. All right. Why would I do that? I don't know. Because I wanted to feel young again. Get the feeling of hangovers without drinking. So. All right. Let's just pull this up real quick. All right, so. Oh my gosh, I was way off, guys. I apologize. I don't know what I was thinking. Okay, so this is where we're at. Why aren't you? There we go. All right, to stop freaking you out. So 10 and 11 is where we're at next week. So unknown results, and then you have a prediction also for what you expect your unknown to grow like. That's due at the end of lab. That's what we're doing. Lab 11, I want to see graphs and descriptions of the results. And then, of course, the third, we do not have lab. That is fall break. The 5th, which is that Thursday, peer review the unknowns, and then you have another week to finish them. So it's actually 3 weeks out. My apologies for freaking everyone out. What's that?
The video?
The video is due 3 weeks from this past Thursday, so yesterday. The 12th. My apologies for freaking everybody out. Suppose I should pay attention to my own schedule. All right. And then we switch rooms. So around the 12th is when we do. Let's start somewhere other than the homework real quick. I'm going to draw up a quick picture. This is actually what I'm doing right now. Upstairs, an experiment running. Time. And then a concentration on the y-axis, a concentration of bacteria. Bacteria typically follow 4 stages of growth. I'm going to put a dotted line here because not always does this occur, at least in the time that we measure it. So this would be a growth culture of a bacteria in a limiting environment. And really what we're going to focus on over the next 3 to 4 weeks is talking about bacterial growth for a couple of class periods. And then we're going to talk about ways that we control growth. So we'll talk physical and chemical growth control, and then we'll spend about a week discussing antibiotics in particular as a more specific example of growth control. What I want you to do real quick with this, 1, identify the 4 phases. So there's 1, 2, 3, and 4. And then secondly, describe, just in words, you don't have to write it down, describe what's happening in each of these phases. I'll give you 4 minutes, 1 for each one. So when a cell transitions from that log phase into stationary phase, what we usually find is that nutrients begin to run low. Something is preventing from these cells continuing to grow in an optimal environment. In our case, in our bulk cultures that we work with in lab, a test tube, what we find is those cells usually are depleted of nutrients. That maybe there's some acid byproduct, and that acid can be growth inhibitory, for example. Typically, the cultures that you guys get when we start out in lab each day is in stationary phase. Okay, so you don't really get to see what's happening beforehand. When we do our experiments in lab next week, we will inoculate E. coli, your particular unknown, and a range of pH values, a range of temperatures, a range of salt concentrations. We'll also measure oxygen requirements, but for the 3 where we're measuring, we're going to do just an endpoint analysis. We're going to look at what was the maximal density of the cells or concentration of the cells at the end of growth. Okay, so we'll really be looking at cells in stationary phase. And typically, again, cells will reach higher concentrations in stationary phase as the environmental conditions are more close to their optimal growth environment. Okay, so there's a lot of fluctuation that can occur here. This right here, that's objective 1. Identify, describe these different steps. Okay? Let's move on to terminology. What I want to do real quick, let's start with this one. So I'm going to write down 4 terms. So mesophile, psychrophile, hypothermophile, thermophile. What I want you to do, talk it over with somebody else, put these in order. I don't care if it's high to low, low to high. And then you don't need to give the optimal and all that for these, but maybe give like a range and that to help support that order. All right, so what's the order? Austin?
Think from like lowest to highest, like temperature wise.
Good.
Drugs can go second as a thermophile.
Perfect. So lowest to highest temperature, psychrophile, mesophile, thermophile, and then hyperthermophile. One that's not included on here are the psychotropes. That is an additional one. The psychotropes usually have optimal growth conditions between that of the psychrophiles and the mesophiles. Psychrophiles grow best at around 4 degrees Celsius. We actually have one. I found it in the freezer over the summer. Psychrophiles have pretty broad ranges. They can grow optimally at room temperature. Some can grow optimally at 30 degrees Celsius. But one of the characteristics of the psychotropes is that they can grow at refrigerated temperatures. Oftentimes our food spoilers are psychotropes. So if ever you've had a slimy piece of meat, it could be Pseudomonas fluorescens, which is a psychotrope. It could be Listeria monocytogenes, which is not only a foodborne pathogen, but also a psychotrope. Which of these is most commonly going to constitute bacteria that we consider to be human pathogens? Mesophiles. Why, Abby? Yeah, that's essentially it. So mesophiles, typically we think that they grow best at around 37, which is roughly 98 degrees, not the band. And so because of that temperature similarity, we find that mesophiles grow best within the range of the human body temperature. In fact, we are never infected, as far as I'm aware of, by thermophiles and hyperthermophiles because our body temperature is too low to even support growth of those microorganisms. Where would we expect to find a thermophile? Nobody's visited any place really cool, maybe in the U.S. that hot springs, that's the big one. Actually, one of the most well-known enzymes in molecular biology is known as Taq polymerase. We use it for a number of for basically making copies of DNA. It was isolated from a bacteria at the hot springs, and Taq stands for Thermophilus aquaticus. Hot water, or hot loving, lives in the water, basically. All right. So this is one set of terms. Another set that I had you focus on were the ones related to pH. I'm not going to review those because I think that actually most folks probably can pick up on those fairly easily. The one thing I'll note is acidophiles versus acidotolerant. There's a slight difference. Acidophiles will only live at low pH values. Acidotolerant organisms are ones that kind of will live maybe slightly acidic, but live well at neutral pH as well and can tolerate the lower pH values. Classic example of these would be your lactobacillus, those that are used to make cheeses and yogurts. The other one that I'm not going to take too much time reviewing right now are the halophiles and the halotolerants. Again, the biggest non-halophiles will grow best at salt concentrations from 0 to 2, maybe 3%. It doesn't mean that they can't grow at higher concentrations, but oftentimes they do so very, very poorly. Halotolerant are ones like Staph epi, Staph aureus, that probably grow best at around 1% salt, but they can handle 0%. They can grow all the way up to about 10% salt. So they can grow over a very large range. The halophiles, higher than that. So some folks mention Vibrio cholerae as a classical example here. Vibrio cholerae is a human pathogen, lives at a minimum, lives in salt water, which is roughly 3.5%. Some halophiles, on the other hand, the extreme halophiles, will live at 30% salt. You change that concentration to be lower than that, they will not grow. So one of the things to bring up here is that there's great diversity in microbes. And we're not only using these terms to describe bacteria, but we're also using these terms to describe archaea as well. Some of those extremophiles that we aren't going to spend too much time focusing on throughout the semester. What I wanted to move on to was to start here with a Venn diagram. I'm going to try my best to draw one. Oh, crud. And we'll label these with our types of metabolism. And you're going, didn't we just cover this? And the answer is yes, but it's still important. So, fermentation. Aerobic respiration. And anaerobic respiration. Are three basic forms of metabolism that we have talked about. There are some organisms, the obligate aerobes, that live only in the presence of oxygen. There are some, the obligate anaerobes, that live only in the absence. There are also aerotolerant, there are also facultative anaerobes, and there are microaerophiles. What I want you to do is place each of those into one of these areas. So if they, for example, perform only fermentation, place them here. If you think that they can perform fermentation and anaerobic respiration, place them here, so on and so forth. If they can do all three, place them here. And this is just kind of, in general, what are your thoughts? Okay? Pay attention to the oxygen requirements and what that means about the environment that they're going to be living in. And that should help you with this. All right? I'll give you guys about 5 minutes to work on this one, alone or with a partner. Yes. All right. Shall we try? Where shall we begin? I'll let you guys choose. Ashley, go ahead. Okay, so where's the obligate aerobe going to, what kind is it going to do? Aerobic respiration. Yep, because, why? What's true about an obligate aerobe? They need oxygen, and we've defined oxygen as a final electron acceptor. Therefore, because it requires oxygen, these ones will not, in general, not perform fermentation and definitely not perform anaerobic respiration. I say in general because we are obligate aerobes, but we do perform fermentation. Just the amount of energy we get out of fermentation is not enough to support our multicellular life. That's why I say in general. So I shall abbreviate if I can, or see if I can put it in here. Obligate aerobe. Okay. Next one. What shall we do next? Obligate anaerobe. Good choice, Erica. What's true about an obligate anaerobe? Or what does it likely do in terms of metabolism? Anaerobic respiration? Anything else? Probably fermentation too, in general. And so when you're thinking about this distinction here, think about which ones require oxygen, which ones don't. And that's how in here at least we'll break this down. Now, there are some exceptions to this. Actually, on the dichotomous key, I'm pretty sure there's one that I had listed that's an obligate anaerobe. It doesn't do any fermentation, though. And so it's one that only would perform an anaerobic respiration. But in general, again, might fit them here. Obligate anaerobe. Okay. Microaerophile. Let's do a microaerophile. Aerophile means what?
Like a minimal amount of oxygen?
Yes, exactly. Emily?
So, like, it can operate with oxygen, or it needs oxygen, but like not as much as it sounds or all that.
Exactly. And that's a great way to put it. So microaerophiles will perform only aerobic respiration. They need it to live. But they're actually killed by high concentrations of oxygen. So they are typically ones that would not be able to survive necessarily on a bench, but may be able to survive like in a crevice or something like that, somewhere where there's a minimal amount of oxygen being provided. So microaerophiles are down with the obligate aerobes. Two more. Oh gosh. All right. Two more. Facultative anaerobes and aerotolerant anaerobes. Let's start with, go ahead, Derek. They are the trifecta. They can do all three. So here we've got our facultative anaerobes. Facultative means basically they can quote-unquote choose. It's going to be based on the environment that they are found in. So these are organisms that would be E. coli, Salmonella. A lot of our pathogens that we focus on fall into this category. Not all of them, but many of them do. If oxygen's present, they'll probably perform aerobic respiration because that gives us that gives them the most bang for their buck in terms of energy yield. If oxygen's absent, they can perform electron transport chain with a different final electron acceptor, and then they also can ferment at least one carbon source. Glucose is typically, if an organism ferments any carbon source, it's at least going to be glucose. How about our aerotolerant anaerobes? Last group. Emily?
Well, I know that it's anaerobic because like they can exist with oxygen, but they like can protect themselves from oxygen.
Good.
So they don't need it.
Good.
I don't know if it needs fermentation.
So everything you said is absolutely correct. The aerotolerant anaerobes tolerate oxygen. They've got, and we'll talk about what these things are that protect them, but they've got molecules, enzymes in particular, that protect them from what we call radical oxygen species or reactive oxygen species. Typically, our aerotolerant anaerobes are going to perform anaerobic respiration. They do also have the ability to perform fermentation. At least some of them do. So they would fall with those obligate anaerobes. So those are our five major ones that we talk about, and just breaking down the type of metabolism that each of them is capable of performing. And again, this is dictated on whether they can survive oxygen or not. We'll add a second layer of this to what allows them to survive the oxygen on Monday. But before we go there, where do these grow in a tube? So one of the experiments we're going to do next week on Tuesday is called a fluid thioglycolate test. And fluid thioglycolate is very semi-solid. It's more kind of like viscous than anything else, not the thickness of the agar plates that we typically use. And thioglycolate is reduced So actually what happens is that as oxygen comes into this media, or as it comes into contact with the media, the thioglycolate reduces that oxygen to water. So it creates actually a gradient of oxygen concentrations. The top of this tube obviously would have the most O2, and as you go down the tube, there's less and less. Okay. Oftentimes these tubes have a oxygen detector in them. It's a chemical known as rosazurin that when it's oxidized, it turns a pinkish color. When it's reduced, it's colorless. So we can use that as well to detect where the oxygen is. Where is an obligate aerobe going to grow in this tube? At the top. Yep, exactly. So it gets localized. An obligate aerobe will be right here. How about an obligate anaerobe? At the bottom. Or basically wherever oxygen is not. So definitely we'll see them at the bottom. Sometimes you'll actually find them if this gradient is really shallow, meaning oxygen's like present here and that's it. You'll find them growing anywhere, maybe like a centimeter and below in the fluid thioglycolate. What about our microaerophiles? Yeah, usually those microaerophiles will grow towards the minimal concentrations of oxygen, so they can't grow right at the top. That amount of oxygen will kill them, but they'll grow just a little bit below the surface. And just a quick side note, microaerophiles out of the 5 of these are the most rare. Not important, but it's rare that we find them. How about our facultative anaerobes? Anywhere, yeah. Facultative anaerobes might, typically what we'll find is a little bit of gradation difference in concentration of the cells. For example, those that maybe quote unquote prefer aerobic respiration over anaerobic, you'll find them growing to a higher concentration, a little bit more dense in the aerobic environment than the anaerobic. And sometimes this is really tough to tell by eye. How about those aerotolerants? Also anywhere. But they, on the other hand, since they don't perform aerobic respiration, their concentration is more consistent throughout the whole tube. As I've described the aerotolerants in the past, they are the honey badgers of those that require oxygen, or those that we categorize for oxygen. Because like a honey badger, the aerotolerants don't give a shit where they grow. With oxygen, without oxygen, it's perfectly fine. Am I speaking to a crowd that has never seen the honey badger video? If so, all of you have homework over the weekend, and I am really embarrassed right now. No, not for me, but for all of you, I'm embarrassed. Okay. I guess you guys might be too young for that. Or were too young for it when it first came out. All right. Let's see. I want to just see if there's anything else I'm missing in here right now. Oh, here are those thioglycolate results. So in this one, Derek pointed out microaerophile just below the surface, really thin layer. This would probably be, based on what I see, an absence of growth up here. This is likely an obligate anaerobe. These are kind of crappy pictures now that I sit here and look at them. I think this one, this is just the flash right here, but I think the growth is only at the top. So this is our obligate aerobe. Facultative, most growth at the top, and then you can see where, these are stab cultures, so where it was stabbed and where it's anoxygenic or without oxygen, there's some growth. Sometimes you'll see this diffuse out if the organism also is motile, but this one is not motile, which is why the results look like that. And then our aerotolerant, equal throughout the whole tube. Okay. Really hard sometimes to distinguish an aerotolerant from a facultative anaerobe, to be completely honest. Okay. Let's do a couple of practice problems. We'll start with the one that's here listed as number 2, so I'll zoom in on it. Okay. So it says an organism grows at room temperature equally well in concentrations of NaCl from 0 to 6%, about half as well at 8%, and does not grow at 10%. It grows maximally at 30 degrees Celsius. It is rod-shaped. It grows better at pH values around 4 to 5 than 7. In a static culture, static meaning not shaken, it grows at a rate of 1 replication per 2 hours. When the culture is shaken, it grows at a rate of 1 replication per 1 hour. Use the terminology we have discussed to describe this microbe. What we are reading here. Ashley. Acetotolerant, so it grows at 7, grows better at pH around 4 or 5 than 7. That would be one way to, oh gosh. That would be one way to define it based on how it's worded. How else might you have defined it? Maybe an acetophile. There's maybe not enough information here for it, but either of those would be appropriate, given at least what we see. What else? It's a psychotroph, and you're using the temperature to help that one. You might say mesophile, but psychotroph's usually a little bit lower than that. I would probably define this one as a psychotroph. What else? I'm glad somebody said that. So it's rod-shaped. It's bacillus. What else? Salt? Probably. Grows for a large range, grows half as well at 8%, does not grow at 10%. Probably what that means is the graph would look something like this. That's a halotolerant organism. Yeah. How would we define its growth based with, or depending on oxygen? How do we characterize this one? So it says it replicates once every 2 hours. In other words, its generation time is 2 hours in a static culture, which would be anaerobic essentially. But in a more aerobic condition, one where we're getting shaking and oxygen kind of constantly dissolving into that media, it's replicating once every hour. How would we define it? Probably facultative anaerobe. The fact that it's still growing at that lower temperature, or sorry, at that in that static culture, indicates that it can perform respiration or anaerobic respiration or fermentation. The fact that it grows better at the aerobic environment indicates that not only is it aerobic, but it's probably facultative. An aerotolerant anaerobe, the growth rate would be the same regardless of whether oxygen was there or not. Okay, 2 quick notes. One of the things that we didn't cover today, but we will be doing with what's happening in lab, is you should be able to identify these based on graphs as well. So in other words, it is not unusual for me on an exam for this to give you a graph and for you to say, define the characteristic of this organism based on what you observe in the graph. Okay, so it's a little bit of a different way of looking at it. I do want you to be comfortable with it. For Monday, the homework assignment is an individual journal entry. It involves math. Okay, it involves some math. It involves powers of 10. It involves some calculations. I am really hoping everybody will try. I am not under any false belief that everybody is going to succeed with it. So try, but if you feel like you're going to cry, please stop. And we'll talk about it some more later. All right? Have a great weekend, everyone. See you on Monday.
Zach?
Yes.